Hello and welcome to our presentation of the gene CSNK2B.
I'm Claire Roberts.
I'm Gabrielle Smith.
I'm Kinsley Travis.
And I'm Maya Van Houten. The CK2 protein is composed of an alpha and alpha prime in a beta subunit. CK2 is a kinase and it regulates multiple systems through phosphorylation. CK2 is an essential protein for muscular development as it phosphorylates the protein MuSK that is important for proper function of acetylcholine. Acetylcholine is a neurotransmitter involved in muscular movement and development. Animal studies have shown that mice with a truncation in the beta unit of CK2 show poor muscular development and have very little grip strength when compared to mice without the induced mutation. Another study done in China highlights the importance of CK2 in neurological development. The study showed that individuals suffering from the variants had less developed dendrites when compared to variant-free peers. This underdevelopment could be a leading cause for seizures experienced by individuals with the variant.
As Maya stated, underdevelopment in the brain is often a phenotype associated with this variant. In the image, you can see where underdevelopment in the cerebral white matter is. This causes severe learning disabilities as well as impaired intellectual development. This underdevelopment paired with less firing of the acetylcholine in the synapse causes varying types of seizures in these individuals. This often presents before the patient is even a year old. This also leads to severe delay in muscle development. CK2 cannot phosphorylate the proteins needed to allow acetylcholine to properly function. This leads to poor muscle development.
Research has shown that this genetic variant is not passed down between family members. Individuals with a CSNK2B variant are usually the first person in their family to have it. The variant is most commonly caused by a missense mutation in the CSNK2B gene. This causes the mRNA strand to stop being transcribed early, which causes a truncation in the beta subunit of the CK2 protein, which leads to improper protein function.
So, what treatments are available? Most individuals suffering from this variant in CSNK2B suffer from refractory myoclonic seizures, which do not improve with most medication. Patients can also experience different types of seizures, and these may respond to anti-epileptic medicines, but myoclonic seizures have been shown to be the most common as well as the most serious, as they generally do not respond to AEDs. Individuals with this variant can also experience developmental delays, intellectual delays, and facial dysmorphisms. Simply put, there are not a lot of known treatments out there for patients with this genetic variants.
This brings us to the research pathway. The first step is for patients suffering from a genetic variant to meet with analysts at PMI to determine next steps for possible therapeutics. These patients usually have a very rare genetic variant. The second step is to create an animal model that has an induced mutation matching the patient's specific variants. In our case, we've been using zebrafish as our animal model. And then the third step is testing therapeutics on the animal models to see if new therapeutics can work to fix the phenotypes of the animals before moving them on to human trials.
Zebrafish work as a good model organism for a few reasons. First of all, they have a large section of their genome that is shared with the human genome. Second, their life cycle is relatively quick, so in a lab, you're able to work at a relatively accelerated timeline. Finally, they have transparent embryos, and so this way you can see if there's any issues with embryonic development as well as other issues that may occur.
The research question we focused on for this class is, can CRISPR-Cas9 be used to cause a double-stranded break in the gene, and does it cause similar phenotypes in the zebrafish models as the PMI patient?
Now we're going to discuss the experiment that we did in class and the methods used. We began the process with bioinformatics. From there, we prepared our samples. The next step involved an in vitro transcription and RNA purification. Finally, the samples underwent a PCR amplification and a nuclease assay.
The first step we used for bioinformatics was to use Ensemble to get the gene sequence in both zebrafish and in humans. We then used BLASTn within this system to compare the sequences to ensure that they had a good amount of similarity. Next, we used benchling to identify good genes and primers. It provided on and off scores to help us further decide if it was strong enough. We also used the Time et al. 2016 paper to have different criteria for the guides. Finally, we use iTaser to visualize our protein. Here you can see our guides and primers, and then also we have a visualization of the gene as a whole with guide 6 and then its corresponding primers. And then this is the protein that our gene produces.
The first step of bench work included preparing the samples and extending the guides. The DNA samples were brought up by pipetting the correct microliters of water into the samples until they reached the proper concentration. This step was done to both the primers and the guides, and the amount of water differed between each one. The next step was extension of the guide sample. This step combined the constant oligo, which was the tRNA that is specific to the Cas9 enzyme, and the designed oligo, the cRNA, that we designed in the bioinformatics steps to make the guide RNA.
Because RNA is needed for PCR, we had to do an in vitro transcription to transcribe the DNA from the extension step. After that was completed, the sample underwent RNA purification to ensure that RNA was the only thing that remained in the test tube. This was done by incubating the sample and centrifuging it at maximum speed. The supernatant was then removed and the pellet was resuspended with water. The RNA gel was run to see if the RNA had annealed, extended, and been transcribed correctly, as well as to see the size of the RNA sample. As you can see from the image, CSNK2B Guide 5 had no results, and CSNK2B Guide 6 had a band at the 100 base pair mark and the 250 base pair mark. There are two bands, which means that there are samples of RNA at two different sizes. The band at the 100 base pair mark is very bright, which could signify that there is a high amount of leftover RNA that did not anneal together. The other band at the 250 mark is most likely the complete RNA guide that we wanted.
PCR was run to determine if the correct fragments were produced during IVT. The fragments from PCR should match the expected amplicon between the forward and reverse primers. If the band is located around the amplicon length on the latter, the PCR has been successful. In the left image, the lanes for guides 5A, 6A, and 6B were all blank, so there are no results. 5B has bands, but they are much shorter than the intended amplicons. In the right image, 5B and 6B are not successful because the bands present are too short. The two larger bands for 6A on the top left add to be the correct amplicon length, but a successful result should have a single band, not two. Primer 5A on the bottom has a single band at 780 base pairs. Although this is slightly shorter than the intended 824 base pairs, it represents the most successful PCR of all of the CSNK2B guides and primers.
A nucleus assay was completed to determine whether or not the PCR product was actually cut in the intended area it was supposed to. The nucleus assay gel was run, of course, by Dr. Crowder to see if the CRISPR-Cas9 could make a double-stranded cut. For CSNK2B, this was done with the 5A and 6A sets. As you can see from the image, we did not have successful results for either set. For CSNK2B5A, there were no bands present for the PCR or nucleus assay. For CSNK2B6A, there were no bands for the PCR, and there was one faint band for the nucleus assay, but this band was not a successful result. Because for a nucleus assay to be successful, there needs to be two distinct bands to show the double-stranded break.
There are several possible mistakes that could have been made throughout this process. One potential problem could have been our amplicon size being too large or our guides or primers not binding properly. This could have been fixed by going back to the bioinformatics steps and redesigning our primers and guides and making our amplicons smaller. Additionally, there may not have been enough extension time during PCR, or our annealing temperatures may not have been ideal. This issue could be remedied by changing the PCR temperature or time. Thank you.